All right, here we go. Kinesiology 230, still chapter 3. We're talking about bioenergetics, and we're talking presentation number 4 here. And we're specifically getting into glycolysis and the glycolytic system to produce ATP. We talked about this before. We've already talked about the phosphagen system. Now we're talking glycolytic. And really, it comes down to the replenishment of ATP. The phosphagen system is very explosive. It relies on these two phosphagens, very fast twitch. We're talking about catabolic reactions. Excuse me. On this side, you're more anaerobic. It's anaerobic, much like the phosphagen system, excuse me. It relies on the breakdown of glycogen in the muscle or the blood to produce the ATP, up to 120 seconds in duration, perhaps running a 400 here, maybe even an 800. You have fast and slow, anaerobic and aerobic glycolysis. And glycolysis itself, it's the breakdown of the carbohydrates that's either stored in the muscle or the blood to produce ATP. Now, during movement, initially it's like the phosphagen system is what ramps up to help us to do any kind of movement, to whether it's throwing a shot put, running a race, whatever that may be. Even if it's a marathon, the phosphagen system is called to action to start that process. During the initial stages, the handoff you have between the phosphagen to the glycolysis system, you have this kind of short burst of movement that happens with one repetition or up to 10 seconds as noted here. But eventually it's handed over to the glycolytic system, and then it becomes the predominant driver for creating ATP. So initially you have like the phosphagen system, and then there's a handoff where there's like an overlap between the phosphagen and the glycolytic system, but then it becomes glycolysis. Now again, this is, remember, it's not as though these operate mutually exclusive from one another. They all work together, where it's a phosphagen, glycolysis, or if it's aerobic or oxidative, they all work to varying degrees, just whether how much they're upregulated or downregulated compared to one another. And then with glycolysis, as noted, it's a high intensity muscular activity that can last for up to 2 minutes. And the limiting factor you have is just how much ATP can be produced. And is it a case where you're blowing through your ATP at a faster rate than you're actually able to create it? So if we look at this, you have the phosphagen system to start off with anything. But there is a little bit of an overlap that you see between the phosphagen system and glycolysis. And there's a transition, kind of like a handing off, if you will, from one system to the other. And you may even have in races where somebody is running and they partway through the race take off and they have a short little burst to try to catch up to somebody else or maybe extend their lead. You might be dipping back into the phosphagen system there a little bit, but really what you're driving at that point is glycolysis. Now this over here, whoa, there's a lot going on, right? And intuitively, when you look at this, you go, man, this is a lot to take in, a lot to process. And I'll tell you, it is. But specifically for our class, I'm going to try to really kind of simplify this whole thing so we don't get completely overwhelmed by it. Glycolysis involves many different enzymes, right? We talked about PFK. We've talked about some of these other ones. We'll talk more about NADH as well. And the striking thing I want you to just kind of take in here when you look at this is hexokinase, phosphorylase. You have phosphate, phosphofructokinase, right? So again, getting back to the etymology of words, when you look at like medical terminology and you look at terminology regarding the human body, a lot of times those are the things that kind of are the tell regarding what they pertain to. And, you get down into, you get past this part of it, right? Whether it's the ACEs or the eights, right? ATE, and you get into here, you have more 8, right? And it becomes an understanding of like, what is going on in the different parts of the system. When you look at this, it becomes a conversation of like the enzymes and what helps the process and what is the end result. The end result, when you look at this, ends up being pyruvate, which is at the very bottom. And ultimately what determines if it's aerobic or anaerobic is what happens to the pyruvate. So you have the first step in this whole pathway, if you will. It's about converting glucose, blood glucose up here, or muscle glycogen up here through either one of these processes into ATP. That's really what you're after. And that's how it's processed and how that whole process happens that dictates and determines what actually is happening. So you have these two main paths. You have slow and fast. And when you look at these two paths, you can look at it and kind of intuitively go, well, if we just draw a line right down the center, we have kind of one series of events over here, one series of events over here. And you're left to believe that it's either like right side or left side, right? And okay, does that make sense when you look at it to have it be like, okay, this is slow, this is fast, or vice versa? And you start to kind of rack your brain on like how to really wrap yourself around what is going on with this monster of a chart. When you look at this actually becomes a conversation where it's not so much right side, left side, but it's more like the center down here and the outside. And when the pyruvate down here at the very bottom, this side or this side, whether it's fast glycolytic or slow glycolytic, when you look at this, one of the key things you have is what happens to the pyruvate. If there is enough oxygen or the exercise intensity is low enough, then the pyruvate goes into what's called the Krebs cycle, and that happens in the mitochondria of the cell. If you have lower oxygen or higher intensity exercise where the person can't actually process out and doesn't have enough oxygen to really process the byproducts that are in the blood, that lactate now, or that pyruvate now becomes lactate. So what you really begin to see with this chart is not right side, left side, like we saw with the yellow and the red. What you see is the pyruvate itself is able to go into the Krebs cycle because there's enough oxygen or the intensity is low enough. If the intensity is too high or there's not enough oxygen to help process, the pyruvate does not go into the Krebs cycle, it becomes lactate. And that's like the main takeaway of everything you see in here. This is key in kind of understanding the whole process. Everything that happens up here, I mean, it has its place and it's everything you're going to learn at some point in your academic career. But for right now, you don't need to freak out about learning these different things and these different steps in here. They're important. Have an understanding that they're there, but ultimately they all distill down to one thing, the pyruvate. And that is the key part. What happens to the pyruvate? Does it end up in the Krebs cycle or does it become blood lactate? That's the end result. And then it becomes a conversation of how that lactate is processed, and we'll talk about that. So here's how you can just break it out. Fast glycolysis, there's a low O2 availability, or there's a very high energy demand. You'll end up with 2 net ATP. You'll have accumulation of lactate and hydrogen ions. So that would be over here to the left and the right, where you have lactate. You take this back over here, slow glycolysis, O2 is available and the energy demand is more moderate, well, you end up with 36 to 38 net ATP through the Krebs cycle in the mitochondria. That's the middle circle here. So if you're able to train yourself to work at higher intensities and be able to recover and tolerate higher workloads and not become anaerobic in nature, or anaerobic, if you will, and be able to stay more aerobically oriented, even though you're working at a high intensity, you can create way more ATP than if you go through the process of fast glycolysis. So here becomes a quiz question alert. I know it, and it's whether it's on the quizzes that I throw at you or what you're going to see later on in your academic careers. If glycogen is used to form ATP, what is the net production of ATP? And glycolysis produces a net of 2 molecules of ATP from 1 molecule of glucose. However, if glycogen, the stored form of glucose, is used, there is a net production of 3 ATP because the reaction of phosphorylating, meaning adding a phosphate group to glucose, which requires 1 ATP, is bypassed. So the question you'll have is, and it really comes down to the difference between an understanding glycolysis right? Glycolysis produces a net of 2 molecules. However, if glycogen is used, there's a net production of 3 ATP. I can guarantee you're going to see that test question somewhere else, 100%. So just kind of understand that there's a slight difference there in nuance in their verbiage and the way they ask the question, but it's all about glycolysis producing 2 net molecules of ATP from 1 molecule of glucose. However, if glycogen is used, there's a net production of 3 ATP. Glycolysis, glycogen. Remember those. All right, signing off, moving on to the next one.